Good morning, everyone. Well, it's morning in England anyway. Yeah, my name is Sarah Solomon. I'm going to be your presenter for today. So we are going to go through everything we need to go through from the June series of last year's exams. If you've got any questions, if you type them in the chat box, I will do my best to answer them. If not, Nash can answer them, and we will go through as we are. Okay. So before we start, I just want to check that we've downloaded the resources and you have a copy of the delegate booklet. You'll find that much more useful to have in front of you during some of this presentation today. So if you haven't already done that, can I suggest that you do that? Okay. Hopefully you can see my screen okay. For some reason, I think my laptop is not doing what it should. So, but we've checked it and hopefully it's fine. Okay. Right. So welcome to today's event. All right. And we are going to be looking through the paper from June. I do appreciate, obviously, we've had another one in October. So bear with us. Okay. But so the idea today is we're going to look at some feedback from the performance of the candidates that sat the exam in the May-June series. I've tried to come at this as a teacher and thinking about what I would find useful in my classroom. So hopefully you'll find it useful in that way. We're going to look at the variation of the standard, the performance that the candidates actually have. And we're going to look at some exam questions and the mark scheme and how that all works. We're going to look at some key points from the examiner's reports, particularly that hopefully will inform your teaching and your delivery with the students and how you communicate this with them. And then if you've got any frequency asked questions or anything and you wanted to type them, go ahead and we'll have a look at some if not. Okay, so let's first of all just have a look at Pearson as a whole. So obviously we are doing Pearson as an exam board, and you've obviously chosen Pearson for a reason, but we are a UK recognised UK's largest awarding organisation, but we do stretch across the world. We have been going a very, very long time, so we are in one of the top, and we are trustworthy in lots and lots and lots of ways. We do sit 3.5 million students a year. We operate in 70 different countries. So thanks for choosing us. And I hope you're having a satisfactory experience. So the international advanced level is what we're going to be looking at. So we're going to be looking at the A-level papers across the whole thing. Now, the first thing to think about is assessment objectives. So one of the best pieces of advice that I was given in teaching was to take the content, but teach it to the assessment objectives. So making sure that the students and the candidates know what they are and how they might be spread across the actual papers. So AO1, that's demonstrate knowledge and understanding of science. So that's your knowledge recall, and that's very much weighted towards the lower end ability candidates. So we're looking at them being able to simply recall the knowledge that they should have learned during the course. Now, AO2 is more difficult. It involves application of knowledge and analysis of evaluation and scientific information that they may not know about. Okay. So it's about taking the content and putting it into new scenarios for them. And the really good candidates are the ones that can do that. So in terms of practicing skills and improving grade outcomes, then if they can do that, that is how they get those top grades. And then AO3, we're looking at experimental skills. So about them having done all of the practicals, not just the core practicals, but the recommended practicals as well. And also to be able to put them again in unfamiliar contexts or into experiments that are similar, but not exactly the same as what they have already done. And you can see that there is weighting here in each of the areas. So you get the percentage in the international AS level, the percentage in the international A2, and then you get the overall IAL percentage. And you can see how the distribution happens. And that's across the whole of the papers. So if we looked at the AS side, you can see that here we have got 3 units, and you can see that the percentage distributions. Note that for Unit 3, there's nothing in AO2A and nothing in AO2B. So it's all knowledge recall and application of scientific content. Okay, so that's the experimental side of things and where it comes in. And then for A level, this is the weighting across the 6 units. And again, you can see that paper 3 and paper 6 are the practical papers, and they have all of AO3 in there. Okay, so AO1 and 2 are assessed in units 1, 2, 4, and 5. Now, when we look at the papers that were sat in June, we can identify something called facility. So these scores on this sheet are about, they are called the facility of the items. So essentially it's what they did get divided by what they could have got. So it's sort of like a percentage, but we don't transfer it to a percentage. The higher the facility, the more they achieved. So the stronger the actual question was. And you can see that we've got quite a lot of questions across the 6 papers that are high in the facility area. So I did only pick out the ones above that were the highest, above 0.7. But the highest ones scored. Multiple choice questions are generally very, very well done. 6A was well done on paper 4. So you can sort of have a look at what the strengths are. Now, these are ones that when the candidates assisting it, they're really good at, and they're well versed at, and they're well practiced at. And we can tell as examiners which ones they are well versed in. Okay. We also can tell what they have been taught and they're not sure about applying. So when we think about statistical data, for example, we often see that they are trying to apply it in areas where it's not really appropriate because they are taught well about statistical data. We obviously want them to apply it where it is appropriate, and they do generally do quite well at that. Okay, so as we get started, our first activity is going to be looking at what you thought your candidates, your students actually struggled with. Where do you think that they found the paper hard or the papers hard? Okay, so I'm just going to give you a minute just to have a little think. And if you want to type in the chat box any of your suggestions, if you want to write on your booklet how you thought your students achieved, then go ahead. There is a space for you to do that on the booklet. Okay, so I'll just give you a minute now. Okay. All right, so in terms of reflection, if we look at the areas for improvement according to the actual statistics on what the papers were, these are the questions that were not so well done, that need a little bit more attention than others. So what we're going to do is we're going to go through these questions, and we are going to mark them as you would, and we're going to have a little discussion about how we might have achieved, okay, on these ones. So I thought it would be useful to look at these command words. So again, another piece of advice is to make sure the students know what the command words mean. So those are the words that we put at the very start of the questions. Those are the ones that are telling the candidates what to do. And the distribution on the papers that we've got, C and C is compare and contrast. Draw, suggest, describe, explain, calculate, evaluate, state, and comment on. All of these questions, all of the command words here were used in the exam. And you can see that explain is really common, suggest is really common as well. So we actually become more common the more occurrences that there are, okay? So when we look at the areas of improvement, these are the ones where the students are struggling, okay? The compare and contrast one is really interesting because quite a lot of the time they actually don't achieve really well unless they actually put this has this and this has this. So they are actually directly putting a comparison down. A lot of the time when we are marking these, they don't get credit for not saying what the other one does. So they might put a whole raft of information about one of the things we're asking them to compare, but then they don't put anything about the other thing. So making sure that they know they've got to do both sides of the argument and both in things that we're asking them to compare is really, really important. Now, the drawing one, this was an interesting one this year because we did we asked about them drawing molecules. And this is something that they really struggled with. And we're going to have a look at a question that shows that. Suggest, this is really common, okay? It's becoming more and more common because it's not really a describe or explain. It's sort of a bit of both, and they're not really given a direct response to actually have. So the suggest ones, they are struggling with quite a lot, and it would be worth spending some time looking at some questions that are suggest and actually tackling those in terms of exam technique. Describe, most of these are knowledge recalls. It's the harder bits of knowledge that they're struggling with there. Explain, we are still seeing them describing and not explaining, okay? And it is really common, and it's been a continual thing throughout the entire sort of history of doing exams. Calculations, there's one particular calculation we're going to look at, and it was, I think it was only worth 1 mark. So, but they did really, really struggle with actually calculating that. And some of the maths, some kids need some help with some maths. Evaluate, there is one here. Again, evaluate, this is where they are putting the data information and not necessarily understanding what the data means. So we need to watch that. State is just a what they just didn't know. So that's a knowledge. And the comment on. And a comment on is a bit like a suggest question. But make sure that the students know these command words and have practiced them. If they practice them, they will get them correct. Okay, so looking at the delegate booklet. So we're going to go through all of these questions that the students struggled with. So all of these areas of improvement, we're going to have a look at 3 questions from each. And we're going to look at what the examiner's report said during it, for it. And then we're going to summarize it all at the end to see what the overall viewpoints are. Okay, so question 3B, this is a compare and contrast question. Okay, so they are, although it's becoming familiar, it's something that the students are still struggling with, the candidates are still finding really, really hard. The question, compare and contrast the structure of the start and stop codons and how they work. Okay, they did struggle with this quite heavily. So there are 3 questions to mark there. I'm going to display the mark scheme for this, which hopefully you can see. And if you want to just spend 2 minutes, go through the questions and put what you would give them in the comment box. Okay, and I'm just going to wait for you to do that. Nash,
Uh, Hi Sarah. Yes.
are you seeing any comments in the comment box? Because I can't see any.
Oh yeah.
Can you?
I don't see any comments here in the chat box.
All right, okay. All right, then I think I'll just go through it all and we'll go from there. All right, so if we go back to looking at what we've got. Okay. Oh, thank you, Cassia. All right, so this first one here, this is worth 1 mark. Okay, so they did really struggle with the idea of comparing the start and stop codons, particularly in their structure. So this is something that we can address in teaching because this is they just haven't grasped that they are different. What they did get, a lot of them, they got a lot of the codons having 3 bases and the similarity between the two, but they didn't get the differences between them. So we've got 1 mark on here, and that is for the both our codons, so have 3 bases, but are made up of different bases. Okay, so they got that mark there. Again, here, they are both codons with 3 bases, okay, but nothing else from the rest. This one, they both contain 3 bases and each code for 1 amino acid here at the bottom, so they get the 3 bases mark. So each of those gets 1 mark, okay? All right, so the second one. So the next one we're going to go through is 6b1. Okay, so this is the one that they were asked to draw molecules, and they really did struggle with this. Now, we don't know if it's because of their knowledge of disaccharides or oligosaccharides, or if they were just unable to figure out how to separate this molecule so that we had those. So we're just not sure. So I'm going to display the mark scheme again so that you can see what it says. And if you could go and do the same thing. So have a look at the mark scheme, and I'll give you just a few minutes or a minute or so to put some scores in the chat box. Thank you, David. Yeah, a lot of them did struggle with this question. And I think a lot of it is because it's so different to the ones that we've done before. So a degree of unfamiliarity with them where we've not asked them to do this sort of thing very often. Okay, so let's go through those. All right, so first one. Okay, so if we look at this, what we've got is we've got two correct diagrams, but the oligosaccharide is the same for both of these. So therefore they would get 1 mark. For this one, this also gets 1 mark. So they've got two correct diagrams. If this had been correct, that would have been 2 marks, but this one doesn't actually exist in our picture, so they can't have that. For this one, we've got issues where the actual things don't touch. So although we've got 1 mark because we've got 2 that could possibly be correct, these 2 don't touch. So therefore they don't get that 2nd mark. I think then this person was probably just being a bit lazy and drawing circles and wasn't actually thinking, okay, this is a molecule, therefore it has to be bonded and therefore they have to touch. And then this one here, this gets 1 mark for the disaccharide because that's correct, but these 2 oligosaccharides are not correct. So when they are looking at this sort of question, should this appear again, when they are looking at it, they need to make sure that when they're drawing molecules that they actually think about how they look, and also don't make molecules up out of nothing. If you're going to break something down, you're going to cut bonds at certain places. That's the core understanding of this question. Okay, so the next one, 7C2. So there's quite a few different answers that we could give on this one. So we did cover quite a lot when the mark scheme was made. The most common ones were marks 0.4 and 6. We're going to have a look at 3 here and stop share and go from there. So here we go. This one. All right, so there's quite a few different options. Nibashi, you can download them on the resource button. So if you download the documents attached, you should be able to download your own copy. All right, so first one. So this one doesn't get any marks at all. Okay, so there's not enough in either of those things to get marks on the mark scheme. When you're marking using the mark schemes, make sure you stick to the words and make sure you know what the symbols mean. So things like curly brackets, for example, means that you've got to see something in them, in those curly brackets. If it's not the exact word, it has to be the equivalent word. And we do try to put all of the equivalent possibles in there, but it doesn't always work. So it's got to be the equivalent word. Also, when you are seeing and, then obviously you make sure that you are seeing both sides of both points within an answer. Okay, so this didn't get anything. So this one gets mark 0.6 for the second statement. So nothing for the word at the first one. Because they don't actually imply that it's not everyone until the second statement. Okay, so they get 1 mark for mark 0.6. And this one gets 2 marks. This gets mark points 2 and mark 0.3. Okay, for those two. Okay, so that's actually quite a good answer. Okay, right. So next question. Okay, so 8C2. So this concept is quite difficult because it's looking at the oxygen dissociation curve, which they find quite challenging in terms of an abstract concept anyway. So we'll have a look at this one for that reason. So going on to 8C2. And I think this is the last one from paper 1. That's right, yeah. So curly brackets are mandatory. Smooth brackets are there to indicate context. So if they are writing it in the wrong context, then you obviously can tell what the right context is we're looking for. Okay, if they don't put a context there, we just assume that it's the right context, but it's just got to be within the right context. So curly brackets have to happen. Smooth brackets are contextual.
Esissima, please come.
Okay, so none of these got any marks on here. So these were all 0 marks. But this is a really good time to bring up about the writing. So if we can't read it, we can't mark it. And it's really difficult sometimes to write, to mark students writing that is really, really messy. So this first one's all right to read. The higher the partial pressure of oxygen, the higher the saturation with oxygen for myoglobin, and then gives an example. Unfortunately, this is not correct. But if we look at this one, this is quite difficult to read. And we will do our absolute best to read them, but it is becoming a little bit more common that we cannot read them. And obviously we do need to read them in order to mark them. So where necessary, try to work with your students. This is hard. I do appreciate it's hard, but try to work with your students about their reading, about being able to read the writing that they give, and get them strategies to write things that are legible for us to read. Okay, so this is quite difficult. I mean, I can just about read that, but it's quite difficult to read. So that gains 0. And then again, this one gains 0. Okay, we're not actually giving anything with the values. Another thing to say, it says, is use values from the graph. Quite a lot of candidates miss things like that. So they miss using the values in the graph, or they'll read the first section and they won't read the section, the second part of the question. They're only reading the first bit. So make sure that they read them. A really good strategy is getting them to underline or highlight or do whatever it is that they need to make sure that they have read the whole question. Okay, so each of those gets 0. Okay, right, so we're going to move on to paper 2. Okay, so paper 2, we're going to start with question 5A2. So this one requires the candidates to apply their knowledge of how phenotypes are affected by multiple alleles for the same gene. Okay, so this is the second question about DNA alleles that has appeared that they have struggled with. So they have some misunderstandings within this in terms of polygenic inheritance. It's quite a difficult concept from my experience. Candidate students that I've taught, certainly, they find it difficult to get to grips with anything beyond monohybrid inheritance. So it's, yeah, they do find it quite difficult. Okay, so same as always. Okay, we're going to go through these questions. I will bring up the mark scheme on here. Okay, hopefully you can see that. And we're going to look through those and see what you think. Okay, just a little note about when we're looking at mark schemes again. So if we've got the word reject in here, that means that we're not going to accept an answer if they refer to polygenic inheritance in this case. We don't very often use the word reject in Pearson. It's quite a rarity, but it is worth noting when you actually are, again, training your students to answer questions, giving them the exam technique opportunities, that if they get it incorrect, but it's so wrong because it's not in the right context, then it's going to affect their scoring. If you see the word ignore, then essentially we're just saying, okay, well, we're just going to ignore any reference based on that mark point. The whole point of this is to give the candidates as much credit as we can. So to give them the benefits of doubts where we can. So we do ignore things. We don't very often reject them. There are others that do it differently. Okay. Right, so let's have a look through those. So the 2 chromosomes. Okay, this one gains 0. All right, and again, we're talking about a single allele, so it's not going to gain anything from that. Now this one just gets mark 0.2, so it is quite a lower end, but they're sort of getting the idea, so it just gets mark 0.2. And this one, again, 0 marks. Okay, so you can appreciate when they are not getting the marks because they don't understand. And again, this is an explain question, so therefore just describing what's going on is not gonna get any marks at all. We need to they need an understanding of it, so we can see where they're not understanding, and the fact that we've got so many 0 marks there does mean that we're not getting that understanding. Okay, so question 5b next. Again, it's a run-on from the last question. And again, they found it difficult, and it's a lot of it is conceptual because they find the whole topic quite difficult. But to spend some time looking at it would be a good guide. And I've seen quite a few questions on genetics, particularly this harder stuff, where they talk about maybe the epigenetics or inheritance of polygenics and that sort of thing. And they do find them difficult. Okay, so let's have a look at these ones. I'll put the mark scheme up. Okay, again, it's quite a long one. Hopefully you can see all of that. Okay. Okay, so this first one is a really good answer. Okay, and this gets marks points 1, 2, 3, and 4. So it does a really nice job of explaining how the gene can give rise to the either a female or a male. So that's a really nice answer. This one doesn't get anything, so this is 0. Okay, they don't have a really good grasp on genetics and how it all works. And then this one gets marked points 1 and 2 on it. So it's sort of a half answer. Quite a lot of the candidates were able to gain a little bit of credit on this, but it's getting them to the maximum scores is proving quite difficult. So that first question, which had 4 marks, was really, really good. Okay. Right. Question paper 3. So paper 3 is the first of the practical papers. The first one that they were really struggling with was the calculation. So this is the calculation question that was really sort of low scored. Okay. So hopefully this won't take long to mark. And I will put the mark scheme up for you so you have it. Three. And within this paper, question 3 was the area that we found candidates really struggled. Okay, so question number 3 there, the answer's at the top. I'll just give you sort of 30 seconds to go through those. And just a note here where it says the accept version, we have noticed that some candidates are writing the times as a dot. Okay, we don't mind that. Obviously this is the accepted standard, but you know, we try to make sure that they get the best opportunities that they can. So okay, so go through these ones. All right, so the first one gains 1 mark. Okay, and they've got 1 times 10 to the 12 bacteria there. We've got quite a lot of candidates that really struggled with this calculation, really shouldn't have done, but did quite a lot with in terms of the indices for the 10. They found that really hard to write it in that way as standard form. So this one gains 0. And then this has got to 10 to the power 4. So that one gains 0 as well. Okay, then we go on to the next one, which is 5B3. Oh, 3B3 rather. Okay, now what we found with this question, and this can be said, I think, for the October series, in my experience anyway, but that is quite personal. This question, they really struggled to give the reasons why they were doing the aseptic techniques. So they found that really hard to give that reason why they had to do it. So try working with the students so they understand not only what the aseptic techniques are, but why they are doing them in the first place. Okay. So stop share that and share that. And there we go. All right, so it's the 3B3 there. So they've got to get not only what the aseptic technique is, but why it is. And another point a little bit more recently, on this one, we did allow gloves on their own. But for them to say sterile gloves is better, because obviously if you just get some random gloves off the floor, that's not necessarily going to prevent bacterial contamination across things. So sterile gloves is better than just gloves, but we did allow gloves on this occasion. Okay. Okay, so here, first question, this doesn't get anything. It's got a Bunsen burner must be lit on a yellow flame, correct, but doesn't tell us why. Kids must always, the lids must always be on if opening the lid. Again, it doesn't tell us why, so that gets 0. This next one also gets 0. Okay, they're not telling us really what's going on. So limited exposure to the air, that doesn't tell us what's happening. Agar should not be touched, doesn't tell us what's happening. Okay, and how to be aseptically technique. And then the last one there, wearing gloves to protect skin from getting infection. We took that as being transfer of bacteria. So therefore we allowed that one for mark point 1, but nothing for mark point 2 because it doesn't tell us why. Okay. Okay, now this last question on paper 3, this was not good at all. They've really found it very challenging to talk about or to answer the question at all about evaluating the usefulness of the data when making the conclusions. So they weren't directly talking about the data. A lot of the candidates actually gave just a conclusion about what they saw. So they were able to say what they saw in the graph, but actually didn't evaluate it at all. So to work on that command word with them would be really, really beneficial because they were not good at evaluating it. So mark scheme. And 3B4. I apologise, I think that's 3B5, not 3B4. I've written 3B4, but it should be 3B5. So on this, the most common one mark point we gave was mark point 4 there. So no error bars, SD repeats. So cannot judge if the difference is if it's statistically different or not, or significant in any way. So that was the most common one. And again, they're well versed in standard deviation bars and what they mean. They generally get that sort of question very correct, and it's done well. So that is the most common mark we gave, but they didn't seem to pick up on many of the rest of these. So this first one here, so this didn't get any marks at all. All right, so there's nothing in there that would be mark worthy. This is a good example of where we do see mark 0.4 on that one. And then the next one as well, we do see mark 0.4 on there. Okay, so we've got no evidence of bars up there. All right, so mark 0.4 was the most common one on that question. Okay. All right, so moving on to paper 4. So paper 4, we are just going to look at a couple of questions on. So 1C2 is the first one. So again, you know, it wasn't particularly great. Explain the positions of the brown and red seaweeds on the submerged rock. Okay, we could take a diagram on this if it was drawn. That would have been okay. We wouldn't mind. We actually generally don't mind diagrams, actually. And if one is drawn and it has the information on it, we will mark it as correct. We're quite happy to do so, to mark it as correct. So 1C2. All right, so just a minute or so to look at that. Yeah, so feel free to encourage your students to draw diagrams, should they find it easier. And I think that, you know, this question was probably more about lack of experience, and it being a bit of a novel scenario than it would have been about anything else. Okay. Okay, so this first one gains 0. Okay, so again, they're not quite understood the relationship with the brown and red seaweeds, the different light and wavelengths getting to the sea floor, so that they would be adapted in different ways. This one gets 0 as well. And then 0 for that one too. Okay. So question 6b2. Now I think I am going to overrun very slightly today. So I do appreciate, obviously, some of you have commitments at 10 o'clock. So if you need to go, feel free. Thank you for attending. And obviously the recording will be available if you want to finish watching what is happening. Okay, but thank you for coming if you do need to go. But I do appreciate I might. Right, so the level-based questions are actually, student perform not so bad on those questions. The essence of it is get them to be using multiple sets of data when they are analysing anything. So in order to gain anywhere near 6 marks, they have to use all of the information given. So if they get that, then that works well. If it's a question that requires them to plan a practical or to talk about a practical procedure, which could potentially happen, then obviously they need to do it in detail, make sure they've got a list of independent, dependent and control variables, what equipment they would use and how they would use it. But essentially, they need to be practicing using multiple sets of data. The most common mistake we see with 6 markers is where they literally do not do that. So they will take the data immediately prior to the question, they will use that, but they won't actually go back in the question and use other data. So if you're going to look at training them on that, you need to get them to do that. That's the biggest place we see is sort of them not doing so well. Okay. And to be fair, on this, and when I looked at the facility, the 6 markers were actually well done in comparison to others. Not the best and not the worst. Okay, so 6b2 on paper 4. Is next. So we're describing conclusions that can be drawn. So this is a bit similar to the conclusions from paper 3, but this time they're just describing the conclusions about the effect of heavy metal. Okay. Doing that. Okie dokie. Yes, Nash, we've got we're over halfway. Well over halfway. Okay, so when we're looking at this question, again, an example of where we might want to when we're marking them, we want to see them having used the data correctly in the right place. So just saying the sentences is not enough. They have to have used the data correctly to get there. So this first one doesn't get any marks. This second one, this gets mark 0.2. And this last one gets 0 marks. Okay. So 7B2. So there could be a number of ways to approach this question. So we'll have a look at this one. So why soil temperature affects the CO2 fertilization effect. Okay, so where we've got the ors here, they can have the context from any of those 3 areas. They can't have a mix and match. Okay, so whatever's the best fit for their answer is where they gain the credit. But we don't mix and match. Okay, I think a lot of candidates in this saw CO2 and immediately just thought photosynthesis, and that was it. Hence why they had lots of references to Rubisco. Okay. Okay. So if, all these 3 again, they don't gain any marks at all because we're not suggesting why. Okay, so no marks for that. Again, we've got a reference to Rubisco there. So no marks for that one, and then no marks for this one either. Okay, right. Got 4 more questions left that we're going to go through. So 2 from paper 5 and 2 from paper 6. So we're not too far from being done. Okay, so I thought it would be good to actually put the question up for this one so that you could see what was being looked at, because it was difficult to judge the answers without having seen that first. So we're looking for them to explain how the structures A and B and their locations enable the reabsorption of solutes on this one. Okay, so answers. we go. All right. What we found with this is that they could sort of say what it was. So mark point 2, they could tell you what it was. But they didn't give any details about sort of where it was, you know, what they were identifying in terms of the position with the kid with the nephron, and then also not always saw the mitochondria's ability to supply ATP. So okay, when we were describing the orientation, we were trying to think of all the different types of scenarios they could give, which is why it looks the way it does. Okay, right, so first question. All right, so this one gains 2 marks, marks points 2 and 3. Okay, so we've got the look at the microvilli there, increasing the surface area, and we've got the mitochondria. This one gets 0. Oh, I think I've done that wrong. This gets mark point 2 only. This one gets mark point 1 and 2, 2 and 3, sorry, and this one gets 0. Okay, question 80. So this is about them understanding why the skin cells doubled. Okay, and they struggled. This was a different way of approaching mitosis here, so they really did struggle with this. All right, so just a few minutes to look at that. All right, so first one here gets 0, so they haven't put anything in it that is credit-worthy. This one gets a reference to cell division, so that would be mark point 6. And this one gets 0 again. Okay, so paper 6, again, question 3 on this paper was the one that was the most challenging to go with. So we are going to look at this. So just to justify a question. Okay, again, they find these difficult. Paper 3. Okay, so 3B3. Okay, so this gains mark 0.1. All right, it doesn't get the control of the other variables such as temperature because it doesn't say why we're doing that. So this only gets 1 mark. This one doesn't get any marks. And this one doesn't get any marks. Okay. All right, so the last question now. So this is question 4A. So it's about preliminary practical work, and anybody who taught sort of coursework legacy would, and we did in the legacy A level, you would realize that we actually looked at preliminary quite heavily, and we might have dropped that off a little bit. So it might be something to pick up again when you're teaching, is to look at the role that a preliminary practical plays in any sort of practical investigation. So last question here. What is the role of the preliminary? Okay. Okay. So this first one here, again, it doesn't get any marks. They've got no credit worthy answers in there. This one will get 1 mark for mark 0.3. So we're looking at how long each takes to germinate. So it gives us an idea of what we would use it for. And then this one gets 0. Okay. So essentially, what does that mean in terms of like bringing that to a conclusion and looking at what that means in terms of your teaching? So there are 2 assessment objectives to focus on with your students to make sure that they make progress and improve their exam technique. The first one is the application with the unfamiliar context. And this, like I said, is a standard one. It's one that happens over and over. It's been happening for many, many years ongoing. So still keep working on that. But then also to look at the analysis and evaluation of data methods, but particularly the evaluation, because they're not able to give what we're looking for in those ways. So try focusing on those. So in terms of assessment objectives. Now, in terms of looking at the examiner's reports and bringing all of that together, these were the points that cover across all of the examiner reports. So this is a whole exam scenario. So the first one, in making sure responses are legible. I said this before, it's incredibly important we can read what they're trying to say. If we can't read it, we can't mark it. It makes it very difficult. The next one, to read the whole question thoroughly, check the answer covers everything, and look at those command words that are in the specification. And a good technique, like I said, underline, highlight, make sure you don't miss any of the points. Next one is to make sure that you look and you experience all of the core practicals and the recommended practicals, and make sure that you can do the IV, the DV, and the control variables for each one. know what validity means, what repeatability and accuracy are, because those are the ones that are coming up time and time and time again. And then in the level-based questions, you've got to use all of the information. That is where we are standing out, and finding that we're not being able to award full marks because we're not asking them what we're asking them to do. They are not doing, they're only accessing the data immediately before the question and not all of the other data that is on there. Show your workings in calculations. Remember that if you put an answer that's wrong, but you've got something right in the calculation itself, then we will give you credit for it, or we will give them credit for it. Also, making sure that the correct significant figures or decimal places are there. If we don't give them, then the common accepted thing is they go one above what we have given. So that's what we would expect. Describing in data, trends in data, or describing conclusions. Describe as a starting point, but then remember to go into explaining and giving the reasons behind what is on. Try not to use the words faster and slower, because we're seeing that a lot. It's better to use increase, decrease, plateau, shallow, steep, but not faster and slower, because they're very difficult to interpret what the students are actually meaning. And be prepared to draw and describe the pictures that we're giving. Okay, so those are the main points in terms of looking at what we want them to improve on in coming series. Now, if you are looking for support, you can go to any of the following areas. And obviously, if you go onto the Edexcel website, you go onto the Pearson website and you look at the specification, you can actually see in there that there are course materials, teaching and support. Have a good look through that, because a lot of that is really useful to see. It's useful to have. There is obviously books that you can buy, and we have recommended ones, including lab books, on the website, so you can have a look through the resources that are there and the recommended resources that are there. Do use this. So Results Plus is a really, really great resource. You can look at your individual school's performance or your centre's performance. You can look at your separate your girls and boys. You can use it as an analysis tool. There is a mock marking service for you, so you can opt into that as well. Results Plus is a very, very good resource to use, so make sure you have access to that. And if you haven't got access, go and see your exams officer and they will give you access to that. And they have this past paper marking and performance reports. You can see how your students did on questions, so you can see your own individual performance and do your own individual analysis on there. Exam Wizard, again, this is free, and you just need to get access to it. So you sign into that and you can make your own exam questions using past papers. They've got the mark schemes attached. Hopefully you know that is there, but that's a really, really great resource. You can download scripts for your individual students. You can do that prior to submitting them for review if you wish to do that, but you can actually see how they have been marked, and you can bring up any discrepancies if you want to. You can pay into these resources. So we've got the curriculum matched student books with active books, with there's teaching hubs. You can choose to opt for those if you want. I've talked about the published resources already. Those are the student book ISBN numbers if you haven't already got them, and where they are located. You've got a subject advisor. His name's Tim Lawrence. He's absolutely lovely. If you want any advice on the subject, you can give them a call and he will get back to you, or send him an email, okay? And they will get back to you and give you the advice that you're looking for. Now, I'm not sure how many people are left, but if you've got any questions, you can type them in the chat box and I will hang around until everybody's gone, okay? If not, thank you so much for joining me today. I do realise I've overrun a bit, but thank you so much, and I shall sign off and let you get on with your day.